The topic is database pipeline and software tools and their application. Our facilitator is Dr. Mariam Idris. She's a PhD scholar and an international partner with Genome.
Hi everyone, I hope you guys are doing really well. First of all, I just want to welcome you all to our webinar. We really appreciate that you have joined us today. In today's talk, I would focus on how to construct a phylogenetic tree using whole genome sequence data via phylogenetic estimation with progressive refinement service. And then I will discuss how to edit your newly constructed tree. Guys, I know it sounds a bit complicated right now, but buckle up. At the end of this talk, I promise you will definitely get the hang of it. For constructing phylogenetic trees using bacterial whole genome data, first we will explore PEPR Resource Center. So as the name indicates, it's a resource center. It has a wide variety of bioinformatics tools and pipelines in it. So we will just use it and we will say thank you to the PEPR people. And And then we will move on to ITOL so that you can edit your tree according to your whims and wishes, the way you would prefer it. Now you may ask me, why are we opting for PEPR and ITOL in particular, since there are so many other options available? The beauty of opting these two lies in the fact that you won't require anything major like a Linux-based system. Now guys, let's be honest. How many of you have a Linux-based system right now? Yeah, exactly. So by opting these two tools, you would be able to handle your data with ease on your own good old laptops. And you would be able to compile the results for your precious genome analysis research papers. So it's more about empowering you guys. So let's discuss the workflow now. First, I will show you how to construct a phylogenetic tree using bacterial whole genomes. For that, we will use PEPR, and there is a service at PEPR that is known as PEPR, and it stands for Phylogenetic Estimation with Progressive Refinement. Later, I will show you how to manipulate your phylogenetic tree using ITOL. And ITOL is another bioinformatics tool, and it stands for Interactive Tree of Life. So right here at this stage, I want to point out that sometimes we construct phylogenetic trees using 16S RNA gene sequences. 16S RNA gene is highly conserved among organisms. But 16S gene is just a single gene. So the trees that you are constructing using it are comparing organisms using only one gene. Now, I'm not implying that we can just that 16S RNA gene is not important. We simply can't deny the importance of 16S gene. It is highly beneficial for identifying unknown bacterial cultures. And in metagenomic studies, the standard metagenomic analysis is based on exploiting these very 16S gene sequences. So if you are interested in constructing trees using 16S genes, I have already covered it in this YouTube link. You just have to go and watch it. And I have used Mega X software for that purpose. With cutting-edge advancements in multiomics techniques, now more than ever, we have a lot of WGS data at our disposal. So like I said earlier, trees constructed using 16S are based on a single gene. On the other hand, WGS offers thousands of genes to examine a phylogeny. So WGS is often considered as a superior way to decipher relationships among various organisms. So now we will just hop onto my browser. But before that, let's see what kind of results are we aiming for. So you will end up with exquisite files like these ones. All right, so let's begin. So the very first thing that I want you to do is search for Patrick. Okay, so this is the address. You need to click on it. On Patrick, the next thing you will do is make an account over here. It will just take a minute or two, but once you will become a registered user on Patrick by making an account, your data would be kept private in workspaces. So only you will have the access to your data. After making the account, I need you to go to the Services tab that is on the top. So just click it and you will see Genomics category over here. Underneath Genomics, you will find Phylogenetic Tree. I need you to go into it. So for building phylogenetic trees, we will first make a genome group. And for making genome groups, we will need bacterial genomes. So how on earth are we gonna get bacterial genomes? For that reason, I need you to click over here and search your organism of interest. Right now, I will search for like T. plantae, Bacillus plantarum, subspecies plantarum. Yeah, that's a long name. So it's a probiotic bacterium. I'm going to search for it. Just wait for it. And I need you to scroll down. And underneath taxa, you will see the organism of interest. And 47 genomes is written here. Just click on it. Now I want you to go to the Genomes tab over here. So you will get all the results here. I want you to just expand it just a little bit more there. So now focus on this vertical green bar. As soon as I'll make a selection, we will have options over here. So the very first bacteria that I want to choose is H43. I'm going to click here and see I'm getting all these options. I can download the genome, I can copy the selected genome, I can view it, or I can just put it in a group. So this is what we are about to do. But first, we have to choose more genomes to make a beautiful, beautiful tree. So let's choose some more. All right. So now I'm going to click on the group icon there. Now, underneath new or existing, you can see this box over here with a drop-down menu. Click on it, and we will choose new group. All right, so group name, let me name it L. Plantarum Genomes. Add. So this pop-up box ensures that your group has been created. Now we will go back to the Services tab and then to Phylogenetic Tree. We have already made the group now, genome group. So just underneath add or select genome group, Click on this folder, go to Genome Groups, and there you can find it. Just click on it and then OK. Now I want you to add it in the selected input genome table like this. Let's go to the parameters. So for output folder, let's make one, create new folder. All right, so I'm gonna write L lantern, create folder. There it is. I'm gonna click on it and then OK. Now we have to write an output name. So I'm gonna write L lantern phylogeny. So leave these parameters as they are. And we are gonna submit the job now. Submitting phylogenetic tree building job. Okay, so now it has been submitted and it would take some time, as you can read over here. So now how will we check the progress of a job? For that, you have to go to the bottom right corner. Just click on the jobs. It's loading. All right. So this job would just queue up over here and it will take some time. So what we will do right now is I will just show you the one that I had already created. So we will go over here and then I will show you this one. Just click on view. All right. So this file, the Newick file, is the file that you would be interested in, most probably. You won't be necessarily interested in all of these files. So just click on it and then view. So that's the beautiful tree that Patrick has created for us with the same set of data that we have just chosen. So you can also view it as a cladogram like that. All right. So now that we have a tree, I will tell you how to edit that tree. For that reason, you will use the Newick file. Click on it and download it. Once you will have the downloaded file, I need you to go to Google again and search for ITOL. There, you got it. So now you have to click on it. Again, you have to make an account over here, just like you did for Patrick. You will make an account over here and you will just upload the Newick file that you just downloaded from Patrick. I had already uploaded it. Here's that file. So I'm going to click on it. So this is the tree. Now you will notice that it seems a bit odd, right? So the very first thing I want you to do is just go to the mode options, branch lengths, and then ignore it. Much better, right? So this is the rectangular form of your tree. Now you can go to the branch options, or you can just, increase the font. You can make it bold or italic. There are so many options over here. So you can choose any one of them. I will go to the advanced tab over here. So bootstraps or metadata. So I will choose display. And I always prefer text over here. So just increase the font. So that's how you can just manipulate your tree. You can examine it and you can just edit it. All right. If you want to display the leaf node symbols, you can do that. Let's make them circular. like this. You can also change the color. I have made them red. There, like that. Internal node symbols, if you want to display them, like this. Again, I'm going to choose a circular one and red one. Choose like this. That's for the outline, and okay, I can choose it as red. So you can just play around your tree. Then the one that I have shown you in the presentation, it was the circular one. You can zoom on it like this. Go to the Advanced tab and increase the font size for Bootstrap values so that you can view them. There, now you can view them, right? And then what you can do is you can also add a background label like this. You can see the difference over here. And then what you can do is you can, this is very interesting. You can just, wait 1 second. Okay. So you can just color code the clades over here. Like that's one clade. And I am gonna go to the editing category and colored ranges, create a new range. And I will just write the name of the range. Choose a nice pastel color. Let's suppose this one. Choose create a range. Then I will go over the other clade. Colored ranges, create a new range. A2. And I will choose a color. Create range. And then I'm gonna go to this one. Colored ranges, create a new range. Like that. And then these two are separate ones, so we are gonna color code them differently. A4. Let's choose a yellow one now. Choose, create a range. And then for the last one, there. So ITOL has a lot of options to offer. Let me fix notes first. Okay. So I think they would look better there. Yeah, it definitely looks better. Okay, so I want you to go to the basic tab again and to branch options and just thicken the lines a bit. Yeah. So if you want to use history in your paper, just zoom it out and then go to the export tab. Increase the resolution. You will have so many options. I'm using the PNG one and export. So the exported tree would have a transparent background. Let me show you once it's exported. Now I can open it. There you go. See, it has a nice resolution. And there's a beautiful tree. So in today's webinar, I have shown you how to make genome groups at Patrick, how to launch a job for constructing phylogenetic trees, how to monitor the progress of your job, the file that you need to manipulate the tree in different programs, and how to edit your tree at ITOL. If there are any questions that you need to ask, just write them below and we will try to answer them. Thank you very much for your attention.